So I think we will go ahead and get started. Hopefully we have everybody here or they'll be joining soon. So my name is Cole. Some of you have met me in the chats recently. I'm going to be kind of like moderating, I guess you would say. And so we're just gonna run through a little bit of business before we introduce our guest speaker today. So hopefully you are seeing the PowerPoint here. We are going to get my computer to work so I can advance my slides. There we go. So you have probably met Marty and Cindy from our Minnesota Sea Grant already, and Jenny and Ann, who is our tech moderator, and Brenna, Lori Hack. we have this ongoing thing, poor Lori. So she is our teacher consultant, and you've probably met us all. If you need any help for anything, please feel free to reach out to us. We're here to help. And all of you now are Center for Great Lakes Literacy partners, and you can learn more about the Center for Great Lakes Literacy at our Seagull website, that's cgll.org. We will also be eventually loading the videos of all of these workshops into our Seagull website. So some of you who have not been able to attend for whatever reason have asked about that. They will be posted shortly, so please bear with us and look for that coming soon. As you know, we've mentioned this before, the workshops and chat content are being recorded for this purpose. So just keep in mind that whatever you say or whatever you share in chat will be available in these recordings. And some of our chat dialogue has been uploaded into our participant Google folder. So if you are interested in going back and following any of those links or reading up about it, it's now accessible to you there, and we will continue to do that throughout the workshop. So we are halfway through our workshop series, and so we just thought we would check in with you folks on a few things. We have mentioned action plans in some of our workshop sessions so far. And at this point, I would like to just take a minute to poll you folks and see where you are and how you feel about your action plans. So I'm just going to give this a few minutes and let folks weigh in. Okay, looks like we're starting to get some responses rolling in. Just so that you all know, we are going to eventually post some more information in the Google Drive about the action plans. So any of you who are feeling a little uncomfortable on that topic, more help is coming. Okay, I'm just going to give it about another 10 or 15 seconds here. It looks like we almost have everybody ringing in. Okay, I'm going to go ahead and end polling now. We have, okay, looks like we have quite a large percentage of you folks who feel like you've got it under control by the end of the workshop series, that you've given it a little bit of thought. Any of you who, like I said, are not really comfortable with the topic, feel free to contact us. We will be putting that information out soon. We'll let you know when that's available. The blank copies of the action plans are available in the participant folder on Google Drive. Also be sure to use the Google Drive resource to learn more about your fellow participants. You will have some time during the breakout rooms for joyful hours to kind of get to know each other. Also, if you want to know more about our staff or guest speaker, their bios are there as well. We have the guest speaker PowerPoint notes available. As I already said, chat transcripts are being loaded there. And we have a lot of other related resources. So if you haven't taken time to dig through those files, take a minute when you can spare it, because I hope you'll find that there's a lot of useful things for you come the next school year. There's also places for you to network and team up with your colleagues on any type of science programming. Let us know if there's other things that you need. One more little piece of business I want to share with you all is before session started, some folks are talking about the leaf pack mini kit that we sent you. This is the week when you would want to set yours out. So if you're going to be participating in the zooplankton last week wrap up on the, I think it's the 19th of August. So you want to have time to set your pack out and get the critters attracted to your leaf pack. You want it out for about 3 weeks. We sent out a lot of great information, but if you lost that information or have questions after reading it, feel free to let us know. So I think without further ado, I would like to introduce Chris Filstrup. He is an applied limnologist at the Natural Resource Research Institute, and he specializes in limnology and harmful algal bloom ecology. So today he's going to share a little bit with us about what causes harmful algal blooms. And I think that will segue nicely. Are you ready, Chris? We're actually running a couple minutes ahead of time today. Yay. I talk fast.
All right. Yeah, perfect. I will attempt to share my screen now, which is something I should be able to do, but we'll see.
Okay, it looks good from where I'm standing.
All right. So is there a way on my screen? Oh, I just need to close that poll. Okay, there we go. All right. Well, hello all. I'm Chris Filstrup, and today I'm going to be talking a little bit about cyanobacteria and harmful algal blooms in general. This is a really fitting topic right now because as the temperatures are warming up more and more, we're starting to see the occurrence of more and more cyanobacteria blooms on lakes. And so these may be hitting the press. I saw there was one story this morning from Wisconsin that someone shared. So this is a really timely topic. And lots of you have probably heard of something called a harmful algal bloom or a cyanobacteria bloom. In the image here, I have a nice picture of one I took from down in Iowa on the lakes I worked on down there for quite a while. But this is what we typically think of when we think of harmful algal blooms, these nice turquoisey green surface scums on the surface of water. Oops. All right, so I actually want to start a little bit differently though, and just kind of reinforce this point that cyanobacteria are members of natural, healthy phytoplankton communities. So this is a conceptual diagram or figure showing seasonal phytoplankton succession or how phytoplankton change through time in lakes. And so this is a temperate lake where we have snow and ice cover here. And once we get ice out, the lake is actually mixing from top to bottom. So you have this period of circulation. And you tend to see things like diatoms that grow better at colder temperatures. They create what we call like a spring peak or a spring bloom. So you see high concentrations there. And then as things gradually start to warm up and the lake stratifies, so the top layer warms up more than the bottom layer, and they become disconnected a bit, and you start seeing things that prefer warmer temperatures, such as green algae and cyanobacteria actually taking off. So that's why this is a perfect time right now, because we're actually in that cyano-y period now. And then as temperatures start to cool later in the season, the lake again de-stratifies or begins mixing from top to bottom. And then we start seeing the loss of our warmer water algae, and we start seeing things like diatoms become more prevalent again. So again, this is just a healthy part of a phytoplankton community. The problem with cyanobacteria blooms is we tend to think of them though when they're a problem, right? So when the system gets out of balance. So this is an image of a girl standing on her dock with a nice cyanobacteria bloom in the background. And this isn't a sustainable part of a healthy ecosystem, right? So this creates a lot of problems. And this is what we tend to think about and why they get so much attention. So from a public health standpoint, so they're really a problem because some types of cyanobacteria can produce toxins under certain conditions. So here's just a beach warning sign from Oakland County, Michigan. So I grew up in Michigan, but so this just shows you all the things. Keep away from harmful algal blooms. Keep your pets away. You wouldn't want to send your children swimming in a lake. I would hope that looks like this. But people do all the time simply because they don't know of the potential dangers. But we're trying to get that message out a little better now. And so the thing about cyanobacteria blooms is they've been around for a while, and there's some really well-known examples of them. So this is the western basin of Lake Erie, which is kind of the poster child for cyanobacteria blooms during the summer. And one of the most, I guess, publicized cases happened in 2014, where what we're looking at here is the drinking water intake for the city of Toledo, Ohio. And this is what the water looked like right outside of that drinking water intake. So back in 2014, there were actual toxins within the municipal water supply where they had to shut down the water supply for like a week. And 500,000 people were without water for that week and had to line up in these big bottle lines. So this gets a lot of attention because of the impact it has on the region. And it is an annual occurrence, but it changes in the severity of the bloom from year to year. The things we're starting to see with climate change, though, is we're starting to see algal blooms in places where we've never seen them before. One of the more local examples here for Minnesota is actually the bloom that happened in Lake Superior in 2018. And so this made the New York Times. It was a big deal because I was actually on the Blue Heron sampling this bloom from offshore. And one of my colleagues who's done work over here on Lake Erie said, Lake Superior now looks like more like Lake Erie than it does Lake Superior, just because we're not used to seeing those blooms. And we're trying to understand what is driving these blooms to become established in Lake Superior. But because so much of our shoreline relies on tourism dollars, this could have a big potential impact on the regional economy surrounding the lake. So why do cyanobacteria produce toxins? I always tell people this is my favorite question to answer as a scientist, because the truth is we don't know. Scientists have been studying toxins since the 1970s, and we still don't know what the evolutionary adaptation is that caused them to start producing these as they evolve through time. Some of the things we do know is that they're really nitrogen-rich compounds, and they're really expensive to produce, so they must have some kind of advantage. But we don't know what that is, and there's lots of hypotheses out there on why they may be producing toxins. One is that they harm competitors or other algae to help the cyanobacteria. They could also have an effect on predators or the zooplankton that eat the algae. So again, minimizing that grazing loss. Some people have said it's to communicate among cells like other bacteria do by sending out different chemical signals. So they know how many other types of algae are actually out there. To protect against different harmful chemicals, there's been some work that shows it improves photosynthetic efficiency so they can grow more with less. And also maybe as a waste disposal mechanism, just to remove some nitrogen from within the cells if it builds up too much. But again, we still don't know, and we're trying to figure that out. So I've been working on a project all summer that's funded by Minnesota Sea Grant, where we're trying to get at what are the triggers of those toxins. And we're trying to do that by looking at the DNA and the RNA of the organism. So one, does it have the genes necessary to produce those toxins? And then we can use another technique looking at RNA to see when those genes are actually turned on. And if we can time that right, we can actually get to what are the factors that cause them to produce toxins. So just for those in the state of Minnesota, so we've been going from Kerry Lake and Peltier Lake all the way up here to White Iron and Tate Lake. So we drive a lot from Duluth. We spend a lot of time in the car, more time than on the lake actually. But I also want to point out a graduate student that I'm working on this project with. This is Kayla Natwara. So this is going to be the basis for her PhD. And up here, this is her filtration setup. So we get out to the lake, she sets this up and filters about 2 liters of water or so. And what she comes out with are these little filters that then she can extract the DNA and RNA. So anyhow, that filter's a little green, and it's hard to tell from the image, but there was an actual start of a bloom occurring in that lake. This is Peltier Lake down south of Duluth. So anyhow, just wanted to share that, and she'll be working on a PhD here at UMD shortly. So you'll probably see her name in the future. To move over, simply what are cyanobacteria? So they're prokaryotes. So basically they're bacteria. They don't have membrane-bound organelles or a defined nucleus. So just like other bacteria you think about. They are photosynthetic organisms. So much like plants, they use sunlight and carbon dioxide to produce a food source. And over here, there's a lot of different forms and a lot of different shapes. And just by looking at these will come in handy during the next part of the session where Cindy's talking about shape and buoyancy a little bit. So just remember this slide when we get to that part. And the other thing is cyanobacteria aren't new. I think people are more familiar and more aware of harmful algal blooms now. But cyanobacteria have been on the earth for about 3.5 billion years. So again, these things have had time to evolve to a whole host of different environmental conditions that typically we don't think about today, which again makes it hard to figure, well, what were the conditions when they originally evolved these toxins to begin with? And this is a image of a bloom. Again, this is Peltier Lake. So if you live near Peltier Lake, I'm sorry, but this is one of the lakes that we studied quite a bit. This is a drone image, and this is actually likely a microcystis bloom. So you can see some of the surface scums. So these things float to the surface and produce these surface scums, but a really pea soup looking lake that we would be concerned may have the potential for toxins in it. And so one of the things I want to kind of get across is that cyanobacteria have lots of advantages and a lot of adaptations to help them succeed. And these are likely to help them succeed under a changing climate. So there's a whole host of nutrient acquisition adaptations. They're very tolerant of difficult physical conditions in a lake that other types of algae have trouble with. They have biological defenses, so resistance to grazing. They have a lot of physiological stress adaptations. For today's talk, I'm just going to talk more or less about the nutrient acquisition part and some of the advantages they have to deal with a changing climate from a physical point of view. So the first thing we know is that cyanobacteria tend to become more prevalent under nutrient enriched conditions. So this is just a figure showing total phosphorus. So in lakes, phosphorus tends to limit growth of algae and plants. Whereas if you think about like your lawn or your garden, nitrogen tends to be that limiting resource. But in lakes, it's phosphorus. So if phosphorus increases, we see an increase in the amount of algae in lakes typically. And what this figure shows you is as that level of phosphorus increases, the proportion of the algal community that's made up of cyanobacteria tends to increase. So we tend to see the most cyanobacteria under the most nutrient enriched conditions. This also plays in with lake productivity. So this is just another graphic showing that as the amount of algae increases in a lake, this is shown by the amount of chlorophyll A. We use that as an estimate of how much algal biomass is there. As the amount of algae increases, the risk of having cyanobacteria blooms tends to increase as well. So that's why we try to limit the amount of nutrients going into our lakes. So what does that really look like, right? So here I'm showing an image of a Secchi disc. This is just a little disc we use and lower down in the water column to see how far we can see it or how deep we can see it into the water. For all these next images, I'm just showing it at a set 8 inches depth. So this is a pretty clear water lake. You can see the nice visible outline of the Secchi disc. And this is what we would tend to think of as a low productivity lake or a quote unquote healthy lake, where we have a really diverse community of the algae that lives there. So as nutrients increase though, the water becomes murkier, it's harder to see as deep, and it tends to green up with more algal growth until we get to our most nutrient-rich lakes. And again, this is that same disc from over here at the same depth, although you can't see it at 8 inches. So within 8 inches deep, you can no longer see that disc. And this is also a community that's dominated by one type of cyanobacteria called microcystis. So what we try to do is reduce nutrients to prevent lakes from becoming this and keep them more towards something more in their natural state. So I said earlier that phosphorus tends to limit growth in lakes, but during certain periods of time, or if phosphorus supplies going into a lake are high, nitrogen can also limit production within a lake. And cyanobacteria have an advantage to that. So right now, this is a Delicospermum colony. So these are the individual cells. And this is what we term a heterocyte, or it used to be called a heterocyst. And what that is a specialized cell that's anoxic, so there's no oxygen present. And the colony can actually fix dissolved nitrogen gas in the water column and use that to grow. And the advantage of this is most algae can't do that. Cyanobacteria have this lone adaptation. And so during these certain times when nitrogen can be limiting, cyanobacteria can grow, but nothing else can because they don't have enough nitrogen. So if this seems a little off the wall to you, we can think about it in terms of like legumes growing, where most people are familiar that legumes live symbiotically with bacteria that can do the same process. They can fix the nitrogen gas to provide that source of nitrogen for the legumes. So do I get bonus points for having peanuts show up in this talk? I don't know. And so the other thing about cyanobacteria, thanks, I got one thumbs up, so that's good. Okay, so the other thing about cyanobacteria is they're anticipated to increase in prevalence in a warming climate. So this is one of kind of the foundational papers by Hans Perl, simply called Blooms Like It Hot. So he got a lot of credit just for the title of the paper. But it says a link exists between global warming and the proliferation of harmful algal blooms. And why this is remember from that original picture we showed, cyanobacteria tend to grow better in warmer temperatures. So there's a couple different ways we can think about this with global change. One is that lake temperatures are getting warmer overall. But for those of us in Minnesota and Wisconsin who have seasonally ice-covered lakes, the other thing we're seeing is the loss of ice cover during the winter and earlier ice out, which also will have implications for the warming of those lakes. So if we go back to our conceptual model here, remember, so this is like just a hypothesized or an example of snow and ice cover on a lake. So as lakes begin to warm and temperatures warm, we're going to start seeing the loss of that. So snow and ice cover will exist for a shorter period of time. Earlier ice out means that the circulation or the lake mixing from top to bottom starts earlier. And also when that lake stratifies will start earlier as well. So what we can think of is this whole community is going to shift towards earlier in the season. So that means the diatoms will have a shorter period of growth, and we'll actually see the cyanobacteria start to occur earlier in the season, which it isn't a good thing, and then they'll persist for longer throughout the year. And just a shout out, a colleague of mine, Kirill, is going to be talking about winter limnology during next week's session. So I'm sure he'll get into a lot more about ice cover on lakes. So if you're interested in this topic, join in for Kirill's talk next week. The other thing that temperature changes is how strongly a lake stratifies. So this is just an image showing you how temperature changes with depth. But basically, in a strongly stratified lake, the top layers of the water are really warm and don't mix with the cold water at the bottom of the lake at all. And certain cyanobacteria have these little gas vacuoles or gas vesicles. Basically, think of them like balloons that they can inflate under certain conditions. And so what this allows the cyanobacteria to do is become less buoyant. So when lakes are strongly stratified and conditions are calm and the water column's not really mixing that well, these there we go. Okay, these cyanobacteria can actually float to the surface, and that's well exemplified over here in this photo where we see these surface scums of cyanobacteria. So what this allows cyanobacteria to do is intercept all that incoming light and shade out anything that can't regulate its position in the water column. So we can think about that similar to canopy effects in forests. So that's another advantage that they have. And finally, I'm just going to end and think about what is the perfect recipe for a cyanobacteria bloom. So if you're concerned about a cyanobacteria bloom before you go to a lake, these are the recipes where we tend to see them become most pronounced. So when we have really extreme rain events, right? So this thundercloud over here with a mean face, we get a lot of precipitation, which leads to a lot of runoff from the surrounding landscape. And with that runoff, we see a lot of nutrients dumped into a lake, which is a potential nutrient source for growth of algae. The thing about cyanobacteria is that they can acquire those nutrients really quickly. They can take them up and grow quickly. So they're able to respond to these big, massive storm events pretty frequently. So if you follow a big storm event with an extended drought period or really calm, low wind, sunny days, hot temperatures, that's when we really start to see these algal blooms start to thrive. So that's like the perfect storm, right, for cyanobacteria to grow in these lakes. So I'm definitely out of time, like always, when I talk, so I apologize for that. But just a recap on what some of these physiological adaptations are. Nutrients, so if there's lots of nutrients in the system, cyanobacteria can grow rapidly and respond to those before other algae can. When systems become limited by nitrogen rather than phosphorus, they can fix nitrogen, so they're basically not limited by any resource. And also these nutrient pulses being dumped into the lake, again, that rapid growth rate helps cyanobacteria respond first. And some of the physical aspects we covered is that cyanobacteria can grow better when things are hot, okay? So they tend to do better, whereas something like a diatom doesn't do as well. And also they can regulate their buoyancy. So as we start seeing things stratify a lot more strongly, they can actually float to the surface, whereas things like diatoms that have these glass cell walls that are really heavy have trouble staying afloat in those types of conditions. So anyhow, feel free to contact me. I'll be around all day. I did want to give a shout out to Elizabeth Yeager. She's an 8th grader who I worked on her Girl Scout Silver Award project on. She had a dog named Barney who died when visiting her grandparents' cottage because it swam in a lake with a harmful algal bloom occurring and got poisoned. And she had never heard about this before, so she made a video to try to communicate with her peers so they're more aware of what harmful algal blooms are. So if you get a chance to click on that, I think she did a great job. So go ahead and check that out, and I'm done. So thank you.
Thank you, Chris. That was actually not bad on time at all. So we're doing really great. However, I would like to turn this over to Lori at this point. She was going to help us monitor the comments in the chat, and we'll see what we've come up with to pick your brain, Chris.
All right.
All right, Chris, thank you so much. Learned a lot about the dynamics of cyanobacteria and some of the other phytoplankton in our lakes. So a few questions from the chat here. Susan wants to know, how does the phytoplankton seasonality relate to the movement of fish?
Yeah, so if we think about like a food web, right? So algae is at the base of that food web. And so normally there's enough growth of algae for zooplankton to eat that, and the fish can start eating the zooplankton and so on and so forth. One of the interesting things though is when you tend to see these communities shift towards cyanobacteria, zooplankton have a hard time eating cyanobacteria, and I didn't get into that. So when cyanobacteria dominate these lakes, less of that energy goes up to the zooplankton, which means less goes up to the fish. So from a seasonal aspect, that's a problem. With the other types of algae, zooplankton are really good at eating them. So a shift from green algae, from diatoms to green algae or so forth, really isn't a big impediment.
Okay, good, thank you. How can we know if we walk up to a lake and it looks like pea soup, if that's cyanobacteria and gonna be harmful to our pets or ourselves, or if it's gonna be just non-harmful algae, just like run-of-the-mill stuff?
Yeah, and that is a great question. So I've talked a lot with Minnesota Pollution Control Authority or agency about this. I always call them authority. So I've talked with them about it, and their guidance is when in doubt, stay out. So if you go to a lake and it does look like pea soup, it may be another type of algae that's growing, but lots of times when you get biomass or the amount of algae that high in a lake, it tends to be cyanobacteria. So again, it could be another type of algae. It could be cyanobacteria that is not producing toxins, but I think the risk of actually going in to that water, you just have to be really careful. Yeah.
Okay, good. Zach wants to know, is a Secchi disc, oh, that was from Joy. I don't know if I said that. Zach wants to know, is a Secchi disc a good way to measure lake health? What benchmarks do you look for when you're looking at a healthy water body?
Yeah, definitely. So the most prevalent way to assess the lake of a health is using a Secchi disc. Nationwide, there's a citizen monitoring campaign called the Secchi Dip-In that I don't know if you guys are familiar with, but you can actually go to your favorite lake, take a Secchi disc measurement, and then report it. And they have data from all over the continental US and likely some from the world as well. Because what that shows is likely how much algae are growing in that system. There's other benchmarks we use, but they're a bit harder to actually analyze as just someone out on a lake. So we also look at the amount of phosphorus in a lake, so the amount of nutrients, right? Because that limits growth. And we also look at the amount of algae, and that tends to be based on the amount of chlorophyll pigment within a lake. But the great thing is all these things are interrelated. So as you get more nutrients, you get more algae growing and more chlorophyll, and the transparency or how deep you can see that disc actually decreases. So they're all related. So again, just by looking at that little black and white disc, you get a good feel for how healthy that system is.
Good. Thank you. It's funny that such a simple tool can be so meaningful. We have somebody calling in on the phone with a 414 area code. If you have a question, you can unmute and ask that to Chris directly. I thought we had something, maybe. Oh. I don't know. Okay. If that person has a question, then, oh, looks like, hold on, go ahead. Go ahead. Oh, no. Okay. Oh, right. Yeah, they might not have a question. Sorry, I'm a little slow on the chat here. It looks like we have a few more questions. Let's just do one more. And then if it's okay with you, Chris, I would love to give you some of these questions and have you respond to them so we can get all the questions answered, that even the ones we don't have time for. Is that all right?
Yep, perfect.
Okay, so one more here. So knowing how harmful fertilizers are to lake health, and really, you know, our drinking water depends on that in some areas, why are fertilizers still legal, and what kind of advocacy would it take to change that?
Yeah, that's a really complex issue. So before I moved to Minnesota, I actually worked in the state of Iowa for a long time, running their whole lake monitoring program, where agriculture is king, right? So it's a really difficult issue, but lots of times now, so remember, phosphorus tends to limit growth of algae in lakes. So lots of times when you go to your store, you buy phosphorus-free fertilizers. In fact, some states, some counties around the country make it mandatory that you buy phosphorus-free fertilizer only.
And Minnesota is one of those, isn't it, Chris?
I think so, but I was just at Menards the other day and I did see some fertilizer with a high phosphorus content for starting things, which I didn't buy, of course. But I think you can still buy it from time to time. But again, if you're worried about your lawn or your garden, most of those plants need nitrogen and they have plenty of phosphorus in the soil already. So you really don't need that phosphorus because all it'll do is run off the landscape. And I think people are getting more aware of kind of fertilizer use and how often to apply it and at what rates. I think that message is getting a lot better. And so, you know, the other thing that can help out as well is having setbacks from the shoreline of lakes and rivers. So I've definitely seen places where people fertilize right up to the edge of their seawall, and likely some granules are going in the lake as well. But if you do have a setback with a little bit of native vegetation buffer, that reduces the amount of nutrients going into the system. So I think there's some practical ways we can work with the existing structures that we have. But again, awareness is critical on that.
Absolutely. All right. Well, Chris, we're kind of out of time here for questions, but I do have some more that we will put in a document and send your way so that our participants can get all their questions answered. So I'll encourage folks, if you still have that last question, send those in and we'll get those to Chris and he can do that. I think now I'll turn it back over to Cole and Cindy, I think.
Yes, thank you. So we really appreciate all those questions and Chris answering them. Now we have time to turn it over to Cindy. She's going to talk to you folks about some ways you can use this information in the classroom and has some great activities for you.
Great. And before, let me see here. Tell me if you see my presentation.
Yep, you're good.
Okay, I gotta get this out of here. So everybody, you know, I haven't gotten a chance to be on center stage yet. And so I thought I'd start out by telling you about one of my kids because I thought you'd think that was fun. So this is Keely. She's a marine biologist and good swimmer. And this is her boyfriend, Scott, also an aquatics person. And his nickname is Sinks Like a Rock. And the reason for that is, you know, I grew up swimming, Keely grew up swimming. We've always been told anybody can float, but Scott cannot. We spent hours trying to help him learn to float. So take a look at this. You know, we told him, fill your lungs up with air. Just fill them as much as you can. You'll float. Nope, didn't work. You know, if you just would gain some weight, because fat is less dense than water, but he's not going to do that. He's very lean. We said, flatten yourself out, spread out as much as you can so you have more surface area. Didn't work. You know, why don't you just use some noodles or something like that and make some appendages so that you have more surface area and can stay up? And he said, no, I want to snorkel. I don't want a life jacket. I want to be able to do surface dives to the bottom of the water and come back up at will. I can't do that, but I'm going to get too tired. So we said, well, swim with us. You know, let's increase our surface area by kind of hanging out together at the surface. There's a lot of safety in that as well. He's a loner. He was gone within seconds. You know, we said, how about self-propulsion? Swim, just keep swimming and you'll be able to stay at the level you want. But, you know, you get really tired. It's very energetically expensive to be having to swim all the time. We thought, okay, sink down. Maybe one of us or maybe something else can come and get you and bring you back up. That's kind of a long shot, right? He laughed. Okay, you know, we were really getting desperate. The only other things we could come up with were, well, we're just gonna put a big bubble around you and you can look at the water from the surface. Or maybe you could find a place where you can snorkel, where there's a big structure at the bottom that you can go into and rest, like some of the things they have down in Florida. You know, and then when conditions are better or you're rested, you can come back out, go swimming again. All pretty silly, right? But phytoplankton have it very rough. So I don't know if you're seeing this thing I'm seeing here. They have to deal with so many things, and Chris just showed you that. And there's so many different kinds of plankton that are able to deal with all those different things. And I'm not going to go into them, but you can see that list there. Pretty intense. Whoops, what's going on here? I'm not forwarding my slide. I know why. Sorry, guys. And we already talked about that seasonal progression. That has so much to do with conditions at various times, as Chris showed you. So what I want to do, knowing that some of you don't have a strong background in lakes, is just step through what it's like in a freshwater lake from spring through fall. So right when the ice goes off the lake, it has either mixed recently or at least it's all one temperature from top to bottom, which means it's also one density. And that means it can mix very easily if the wind's blowing. Lots of oxygen typically if it's mixed recently. There's exceptions to every rule in this science, so bear with me. The sun starts really warming up that surface layer. The wind comes out and mixes that warm surface water down. But as water warms, of course, it becomes more and more buoyant. The density decreases. And that's what Chris was talking about with summer stratification. You've got warm sunlit water over buoyant, or warm sunlit and very buoyant water over cold, dark, and dense water. So, you know, algae need to photosynthesize. So they are going to be restricted to that upper layer, and they'll be competing for space. They'll be trying to avoid predation, et cetera, et cetera. And as time goes on, one of the things that we are going to really focus on with your activity is they're also trying to avoid sinking. And unfortunately, just it's inevitable that they will eventually sink down to a point where they can no longer photosynthesize. They'll die, they'll get eaten. It's a great area right there at the bottom of that upper warm layer for fishing, and there's so much rich activity going on in that layer. I'm not throwing all the technical terms at you though. So, everything I showed you about Scott and Keeley is absolutely true with algae. So the lesson that we're aiming towards is all about ways to slow your sinking, or to basically outcompete other organisms in the water. So, you know, we talked about holding your breath, and Chris just did a really great job of describing gas vesicles, and that especially cyanobacteria actually can regulate their buoyancy really well. Diatoms, those poor things that, you know, they're a lot heavier, they do best when the water's really turbulent, because turbulence helps hold them up. They also can have oil droplets, and oil also, just like fat, is less dense than water. Oil basically is a fat. Increasing your surface area. A lot of algae have very flat cells, very spread out, so that their surface area to volume ratio makes them be much more able to stay up in the water column. Many different groups of algae have representatives that have spines or spikes or other sorts of appendages. And again, that's partly in some cases to avoid predation, but it's also for helping you stay up by increasing your surface area. The colonies are gorgeous. So here's an example of Asterianella, which is a diatom, and then a green alga. There's tons of examples of algae that form colonies, and that again is for many reasons, but one of them is to keep it buoyant. Self-propulsion, we already talked about blue-green algae, or we didn't talk about blue-green algae, but there's a whole class, dinoflagellates, where there's a number of them that can move themselves. They basically can slowly move through the water to adjust their buoyancy. Couple examples here. And this is so cool, and I just learned about it in a lecture really recently. There is a blue-green alga, and Chris had a picture of it earlier, Dolichospermum, that can actually hitch a ride with a protist, another kind of organism, Vorticella, to get back to the surface once it's sunk too far. And it helps it outcompete some of the ones that really can control their buoyancy really well, like Microcystis. I thought that was so cool. I put the citation in because I was afraid Chris would argue with me. I didn't know if he knew about it. And then there are a lot of algae that have some sort of a gelatinous sheath around them. And that again would protect them from some predation pressures, but it's also increases their buoyancy. And there's many, many types of algae that have some sort of a resting stage. So yeah, they're eventually going to sink and end up at the bottom or get eaten and turned into nutrients again. But a lot of them can have a resting stage that can stay actually in the sediments for a very long time, so that when the lake mixes again, which means all the water is getting stirred around, they can end up back up at the surface and have a chance to live again. So all of that is a precursor to the lesson. But before I get there, I wanted to tell you about this website. I think it was a graduate student that put it together, but it's a clickable key to go in and look at different types of algae that are resident in Lake Superior. So I thought you might want to know about it, and it'll be in the slide collection. So the activity, which is in your folder or in your folder on the drive, is the Great Plankton Race. And many of you have probably heard of this. It's having kids do some hypothesizing and then create their own plankton to be able to have races with each other. Of course, the goal of the race is to sink slowly, and they can't float on the surface. And the reason for that is a lot of algae cannot handle the intense light at the surface, and they may be more subject to predation by other things and other reasons. So you want your algae to be as neutrally buoyant as possible, but they should be slowly sinking. They have to have washers in them as weights because they need to have their organelles and all their other things that they need for physiology and reproduction. So just a few pictures. And then I wanted to tell you that there's 3 different versions. This lesson is all over the place, and most of them have been developed for marine systems, but they work perfectly for freshwater systems. Just looking at, I'm at 10 minutes, okay. You can use these. You see I have one of them listed as being for younger students. It's just a really simplified version of it, where maybe making the plankton and talking about surface areas as far as you go. The one at the bottom, the Great Plankton Race with discussion questions, has some really good thought-provoking questions in it. So these are just different versions of a similar thing. The Plankton Races that I labeled More Science Concepts has way more connections to the real science, like surface area to volume ratios, the math that you could do, Reynolds numbers, which we're not talking about. Just trust me that for little tiny organisms in the water, it feels like they're swimming in molasses, which there's even some options to do the experiment with corn syrup or other things that demonstrate that. Buoyancy, density, temperature and density relationships, all those things are described. And so in my mind, this could be used with any age from very, very young up to college students with the right frame of reference and challenge. So that's basically it. I just wanted to fill you in that on this website, on this, PowerPoint presentation, I've added a whole bunch of other information. I just stuck in the Secchi dip in. There's some citizen or community monitoring program stuff here, a diatom video. I may be adding more to this, so check it when you get access to the slides. And then there's some more stuff from a Water on the Web website that has all kinds of really basic information and some other things. And so that's what I had to show you. Also, there are other lessons on the website, and some of them are pretty cool because they involve creating little microcosms of ponds. And then you can, doctor them up to see which ones turn greener with various things that you put in them. So I encourage you, take a close look at what else is in there. And that's all I had. So I'm going to end the slideshow and see what you're all doing.
Okay, thanks, Cindy. We're actually doing really great on time today. Yay! So normally we would take our break at 5:55. So we're just a minute before that. I think unless Cindy or Ann or Marty has anything they would like to add, then we'll maybe just push forward with break. When we come back, we will go into breakout rooms for everyone to be able to kind of talk about the lecture and the activity lesson plans that Cindy was just sharing with you. We're going to use that bit.ly link that we've been using in the past. So if you can pull that up when you come back from break and have it handy, that would be great. The link is also added in the chat. So if you'd like to go ahead and unshare your screen for me, Cindy. Okay. All right. Well, please have a nice 5-minute break, and we will see you all when we come back. If Ann is ready, I think we'll probably start sending you folks your messages to break off into your breakout rooms. We just wanted to point out that the correct bit.ly link is the 20 sturgeon link. So there was a little confusion. I had the wrong one on the slide originally, but 20 sturgeon is the correct link. Whenever you're ready, Anne, feel free to start directing everyone out to their breakout rooms.
Yeah, and just a reminder as before, you are to pick a recorder. You have a facilitator in the room with you, and then also to pick out someone to report out when we return at the end. So I'll send you a 5-minute reminder that the meeting will be, or your breakout will be ending. And then for some reason, I can't seem to remember to make it 60 seconds. So you'll get a 15-second reminder that the room is closing and you'll come join us in the main room. So have fun, enjoy, and enjoy your conversation. See you soon.
So we'll just give everybody a minute to get home. And then what we would like to do here is have somebody from each group kind of just spend like a minute maybe. We do have to be mindful of our time, and just share some of the key things that you discussed in your group, or the things that were most interesting to your group. So Anne will have to fill me in. I forget how many breakout rooms we ended up having, but group one?
There were 4 groups total.
4, okay. So if we'd like to go ahead and have somebody volunteer from group 1 to share out a little bit on things that you folks found most interesting, or questions.
So I'm going to do that for our group this week. We had some difficulty getting everyone in at the same time. So one of the ideas was an art teacher doing tessellations, similar to the format of the clusters of the algae, and working in some of the math with that as well. Another idea was using the graphics that were in Chris's PowerPoint related to the Secchi discs that show the different depths. So actually using those images and then having graphs that represent increasing levels of cyanobacteria or phosphorus, and having the students match the graph to the actual visual. Another idea was talking more about how our activities impact the environment and the phosphorus levels, doing consumer labels off of phosphorus packages. We did talk a little bit about the intersection of the zebra mussels making water clearer. They apparently don't like the cyanobacteria so much, and that the fact that the water is clearer, we can now see shipwrecks that we haven't seen before. And we talked a lot about safety, collecting samples and how to do that. And the basic message was you don't need to be overly concerned to use basic hygiene of washing your hands and not putting your hands in your mouth and that kind of thing. So that's a quick synopsis of what we talked about.
Great, that's some interesting things there. Who is in group 2? Would you like to share out and see what you came up with?
I think that was our group, but I'm not sure what number we were. I was with Marty. Are we group 2?
I believe so, yes.
Okay, sorry about that. We had some really great conversations about some of the standards that were met, or we could continue to move on based on this discussion about adaptations, and specifically form and function and how they're related. So it was really nice to see that. Also using and making models. We also really liked how this activity could really lead to a lot of like family science building time. So, you know, the Great Plankton Race could become a family activity, but also creating your own homemade sampling equipment and using that as a group. So.
Okay, great. Thanks, Carly. A lot of different things. So we're getting quite a variety today. That's awesome. So now group 3.
I think that's us. And so we talked a little bit about adapting the Plankton race for virtual learning by using like quarters instead of washers. But then we came up with a really awesome lesson that's just amazing. We talked, and it's really good. We talked about starting with that video, so it's a real world phenomenon, so they kind of have a storyline and get the kids excited, and then giving them, if they're virtual, or it doesn't matter, they could do it face to face, but giving them a data set of how algae changes in different lakes and having them analyze it for different temperature or different type of lake. And then having them think about the, what's the future going to look like if nothing happens? Looking at human impact, how could human impact affect that? And then having them create like an advocacy or some sort of a communication piece that they would, okay, so now you found all this out, how are you going to get your message out to people? So that could be, could be a tourism thing or whatever, but this would be, it'd be a phenomenal like cross-curricular lesson. So
yeah, for sure.
I got excited.
Yeah, getting the kids to talk about what they did is important too, right? So
yes.
Excellent. Very good. So these are some really exciting workshops or some breakout rooms today, it sounds like. So we just have one more group, group 4. Do we have a volunteer to share out? And who was it? Do you have a record of who is in
the school group? I'm not sure what our number was.
Yes, we need to, we'll make that clear next week. Thanks.
Yep, that's us, Michelle.
Okay, great. So do you have a few things to share with us, Michelle?
Yeah, sorry, my tech was glitching there for a moment.
No problem.
Okay. So we were mostly high school teachers. In terms of adapting to the classroom, we saw that this would be pretty easy to go into like a wildlife class or an ag class. Understanding runoff and yard fertilizers, and understanding that, agriculture is often blamed, but it, on a per acre, homeowners are way worse offenders than farms typically. And then also just that plankton race thing would be really fun to integrate with high school kids, with a lot of cross-curricular ties from physics to biology and kind of that STEM approach. So we liked that. And then we talked quite a bit about online hybrid that would, or home schooling, home learning, that plankton race you could do. I think another group said this, although we were like, you could use pennies for weight. If they were post 1982, so they're all roughly the same weight, and that you could use whatever materials, and that would be good for learning from home, and that you could probably do data-based labs with student investigations on their own questions with buoy data and satellite data, kind of tying to previous weeks with what we looked at this week. And then we kind of got off track talking about our action plans, which range from, co-curricular aquaponics to food webs impacts from invasive species and microplastics.
Excellent. Wow, I'd say there's a lot of different things going on this week. So thank you so much for sharing. We are kind of running short on time. We want to give you folks a break before those of you who are returning for joyful hours have to get started. So thanks again, everyone. Thanks to Chris and to Cindy for the great information today. If you have any additional questions, please feel free to contact us. And we will see you folks back in about 5 minutes. My computer says it's 10:28, so we'll be aiming for about 10:33 when we come back. See you then. Thank you. So for those of you who are coming back from break, thanks for joining us for the Joyful Hour. We will be getting started here very shortly. Anna is making the final adjustments to our breakout room rosters. So shortly you will be getting your message there to join in. Just like in previous weeks, we are going to be using a Google form to record your discussion. So the link for that is in the chat. It's the 20 Dragonflies bit.ly link. Somebody in your group, if you would please do the recording of your discussion, and then have somebody who is willing to share out at the end of the joyful hour when we all come back to the home main group. And so this week we're talking about the research vessel Lake Guardian. And so in your rooms, you'll have your facilitators who will share some of their experiences and be open to asking questions and seeing where your joyful hour takes you. Any questions or concerns before we hop over?
For those who have more questions after the breakout, if you are interested, there's also information about the Lake Guardian on the Siegel website, and I can put that link back in the chat. We've had it in our message board several times. It gives you an overview of what that opportunity entails.
Thanks, Cindy. I'm sorry, Jenny. Here they come. Welcome back.
It's fun to see everyone's head popping up.
All right. I think that looks like everybody. So welcome back. Hope you had a great joyful hour session. So we have about 14 minutes or so to kind of debrief. We only have 2 groups. So that gives each group a little bit more time to talk. So if we'd like to get started with group number 1, somebody who would like to share your experiences in your joyful hour.
Sure. So it's Kristen. I'll
Hi.
go ahead and feel free for the rest of the group to chime in. So some of the highlights were the relationships formed with the fellow teachers. So having the opportunity for this intense week-long experience and getting to know fellow teachers better from across the basin. So Kathy had mentioned that she still stays in contact with some folks, and they even designed some projects together and presented at a regional conference together. So, you know, having that kind of support was a nice benefit from the experience. She also found that it was easy to reproduce what you learn on the ship to bring back into the classrooms. So it inspired her to get a grant to take students out on the Dennis Sullivan, the schooner, and also do lessons in the classroom. And MJ was asking about, is it only science teachers that can be on the ship? And we talked about how we do welcome interdisciplinary teachers. We have had, you know, math teachers and English teachers, special ed. So it's not just science teachers, although it's primarily, but that's a good way to help expand not only the art teachers knowledge base, but then also the science teachers who are on the ship if they have an art teacher on board to help them think about things differently. And also just John and Carly had mentioned, you know, the whole thing of working with real scientists. So you're working side by side with them. You're doing the research. So you're really getting a firsthand experience of what it's like to do science and feel like a real scientist. So you're collecting samples. You're spending a lot of time in the lab working up those samples and then with the data. And so it gives you more ownership, but it also just makes you realize like how tedious the process can be. But that it's, you know, it's just good to experience because that's just part of doing science. And Carly had mentioned, and I think John, you probably, yeah, John, you had also connected with scientists afterwards so that they're really approachable. And that's, you know, part of what we're trying to get across on these these cruises is that, you know, science is not the ivory tower. Like it's the scientists are very approachable and they love to share, you know, all their knowledge and also learn from you guys. I'm sure I forgot something. Oh, we talked about microscopes and a way to be able to have students have some of that experience if they're not going to be in the classroom. And so Kathy had mentioned fold scopes, which is a more inexpensive way to get microscopes out to the classroom. And I'll let Kathy, you know, explain it more. And then Ginny had mentioned pluggables, and looks like Nicole has something. So, you know, different options to give that students experience. Oh, you've got Foldscopes. Awesome. So yeah, so anyone else want to chime in?
Are you trying to say something to us, Kathy? Oh, okay.
She did a wonderful job.
I just want to add one thing that came up at the very end, and that was Carly also talked about kind of using the scientists afterwards in her classroom. They had an opportunity to ask the scientists and direct questions from the students as they put their own research projects together. So that was one other aspect that she mentioned.
Very cool. Excellent. Well, thank you for sharing. That sounds like a really fun group. So let's turn to the second group and see if we had some similar or different experiences we'd like to share.
Yeah, we certainly had some similar discussion as the other group. We also talked about because there were a fair number of us who've had some type of shipboard experience. So we talked a little bit about, you know, if you're going to apply, what are some of the things that help you make a really strong application? Like talking about how you might use it in your classroom and share it with your colleagues, those types of things, which are really helpful. And then we went through a little bit about what, as a teacher, did you take back to your students? And how did it kind of affect your classroom on the other side? So Michelle in particular talked about like the labs and the research that was done on the ship. That really helped her bring that back to her classroom. And I would guess just feeling more confident about doing those things as well. And that Lori was talking about how it really, even though she had had previous experience, it also made her excited again about really getting her students involved in that and connecting them with the work that's going on. So she started doing more of those things in her classroom in the spring. And, you know, one of the other things is that with doing something like this, you may be on a ship with teachers from across the basin, which really allows you to connect and collaborate with them in different ways. You know, those people are now people who are part of kind of your network that you can call upon to help out or ask questions, or even just connect your students to, you know, a classroom in Minnesota or a classroom in Ohio, depending on whoever you're connecting with. We talked a little bit about the difference between sailing on the Sullivan versus the Lake Guardian, much less the just differences in the experience, kind of in general, of where you sleep and the food and that kind of stuff, but also on the Lake Guardian, the focus is really much more on the science and doing research yourself, and really you leading through that. The Sullivan has a different focus, you know, so there's a little more of an overview kind of focus, not that research focus as much. But that sometimes, while these experiences are amazing for teachers and it's really great to have them, sometimes it's also great to have experiences either on the ship or just in general locally, so that there are, you know, the Sullivan for students and teachers in Milwaukee, that's a really great resource because they can just go out for 2 or 3 hours, and it's, you know, a big group of students can go and all of those things. So that can really get them to be out on the water. But there are other youth ship experiences out there. So that's kind of a great opportunity that you can check with. And some of the people from Sea Grant did mention that at times there's potential for mini grants through Sea Grant that can help pay for those types of experiences. So that was kind of another chance to do something. So then we kind of ended, although we didn't really get through everyone, we ended up talking about what as a teacher really got you interested in water, or what, you know, what kind of brought you to learning more about it. And that's, you know, kind of a good, just thinking about that even kind of gets you thinking about how to get your students to that aspect. So anyone else can continue to add to that though.
Thank you, Liz. I appreciate that. There was some interesting information I didn't know. I didn't know there was that great a difference between the focus of the two ships. So fabulous. So I guess we have a few minutes left. We kind of wrapped up really quickly today. So is there anything that anyone from our workshop team would like to share with the group before we turn it over?
If anyone is interested in applying for the Lake Guardian, assuming that it will sail next year, I think that we'll be looking for applications starting December 2020 or so. Does that seem about right, Kristen?
I would say probably closer to January or February.
Okay.
Okay. Well, thank you everyone for sharing. We do really appreciate your great conversations. I don't know about anybody else, but I'm sitting here looking at about 11:27, so we actually have a few minutes left if we just want to open it up to any random questions anybody has, or any random comments anybody would like to make. Otherwise, I think we have had a nice full day.
Perhaps Kristen can address the comment in the chat about people returning on the Lake Guardian. If there's opportunities to go a second time.
Typically not as a teacher, but if there was room to go as a staff, it just depends how many Sea Grant staff members are going. Just because it's only 15 teachers a year. But we did have, like when it was on Lake Ontario, we were able to have a former teacher come out because it was just myself and the New York Sea Grant lead. So we had space for one more person. So it kind of depends on that year.
Great. So there has been some comments in the chat about the foldable microscopes and other inexpensive microscopes. So if you folks are interested and haven't had a chance to look at the chat, those notes will be posted into the participant folder.
Cole, Cole, I was just going to add that I'm also putting them into that PowerPoint I talked about as more resources. So,
oh, fabulous.
So definitely look for a new version of it in a couple days.
Okay. Yeah, so I'm just kind of scrolling through here to see if there's any other important points in the chat. And it looks like you had asked about, are there other opportunities in your communities to get students on the water? So I don't know if anybody has been monitoring the chat. Does anyone know of any other types of opportunities besides using the Dennis Sullivan?
Well, we've been working with a group here in town called Wilderness Inquiry, and they get kids out on canoes that hold like, I think 10 kids or 20 kids, something like that. They're huge canoes. And many of our kids have never been out in a boat. in St. Paul. So it's really a great experience for them to get out there, and it's super safe, and they're professionals, so they don't have to worry about all that. They tell them they prep them with rowing and wearing life preservers and all that stuff. So really a very positive, awesome, usually a day long. It's usually most of the day, and they have other stations going on. They're kind of pricey, but for one thing, you can get grants to help pay for it. Or actually, a year or two ago, I got a Seagill grant, only $500, but we used that to partner with them, and they picked up the rest of the cost. So that was really awesome.
Excellent. Thank you
Yeah.
for sharing.
Mm-hmm.
And Marty did put a link to that in the chat for anybody who wanted to follow up on the wilderness inquiry.
And they're a Minnesota company, and they're based down in the cities, but they have made it as far north as Duluth before. So they do travel. I don't know what the range is, but there is some travel capability.
Yeah, really neat.
Well, thank you, everyone. It is 11:30, and so I feel like it is our duty to say goodbye and thank you. We hate to see you go, but hopefully we'll see most of you back next week.
See you next week. Thank you.
Good.
Bye.
Bye, everyone.
Bye, everybody.
Thank you.